Hi everyone, this is Edith. Today I'm going to show you how to calculate the sample size for a group project. This is a different calculation than the one that is actually on the final draft, but I think it's a good example of what happens when you change a little bit the data. So first of all, we have to remember that we had a lot of trouble calculating sample size because there is not enough literature about our topic. So when we were trying to find articles, we couldn't find one that was using curcumin and piperine both in COPD patients and that was at the same time using the same primary outcome that we have, which is annual rate of exacerbations. We tried looking for articles that were using both curcumin and piperine in COPD patients, but we also couldn't find that. So at the end, since we were very worried about this, we tried to look for articles that would be representative of our population, which is COPD patients. So we searched for articles that were only using COPD patients and that were having the same primary outcome that we did, which is annual rate of exacerbation. So just take that into consideration right now. Remember that to calculate the sample size, we need to take into consideration power, the variation, which is the standard deviation, the alpha, the effect size, and the expected dropouts. And just for a friendly reminder, because we're going to be using this, I put you here this data formula to calculate the sample size. So the first step is defining the effect size. And honestly, we had no idea of what a good effect size would be. So we looked for what other researchers were putting on their articles. We decided to look for articles, as I already mentioned, that we're using our same primary outcome and we're using our same population. And we found 3 articles, and the 3 of them were actually trying to detect a 15% reduction in annual rate of exacerbation. So, since these 3 articles were very important articles and they were all about our topic and they were all including a 15% reduction, we decided to use 15 the same way in our article. Just for you guys to know, the first option, which is the one I'm putting here in pink, this is the one we chose in our final draft. And as you can see, they're calculating a sample size to detect a 15% lower annual rate of moderate or severe exacerbations, which is the same thing that we will be doing. The second step is defining the mean and the standard deviation. Now, this is a point where I'm playing with the data, and this is the part that it's different to our final calculation that we are doing in our draft. So here is just for you guys to see what happens if we would have taken a different trial into consideration. So in our final draft, we are using the New England Journal, which is triple therapy, to calculate the sample size. In this example, I'm using a different trial, which is called the benralizumab for the prevention of COPD exacerbation. Now, using this article, I took this information, and let me show you where it is. So we go to the article, which is right here, and this is the article, benralizumab for the prevention of COPD exacerbations. As you can see, this article is also testing the efficacy and safety of this intervention, which is benralizumab, for the prevention of exacerbations in patients with moderate to very severe chronic obstructive pulmonary disease. Pretty much they're doing the same thing we are doing, but with benralizumab. So we want to take a look at their mean and standard deviation. So we go to table 1, here it is, and we can see that this article was having 2 different trials at the same time. They have one trial called Galathea and another trial called Terranova. The important part here is to see that this one has 2 interventions and 1 placebo, and this one has 3 interventions and 1 placebo. So we just want to get their mean and standard deviation. So we go to the primary outcome, which is the one that we're interested, and it's the number of exacerbations in the previous 12 months. In here, we're going to take their data. So their control, which is placebo, has a mean of 2.35 and a standard deviation of 1.42. This will be our mean 1, which is control. Now, for the intervention, they have 2. So we're just going to take 1. We're going to take this one. And here we're only going to take their standard deviation, which is 1.17. We're not going to take the 2.31 because we want this mean to have a 15% reduction rate. So you're going to see what I did here. So their mean one or control is the one that they were stating, 2.35, and is the one of the placebo. And the standard deviation is the one that they mentioned. Here's where the magic begins. For the mean 2, we calculate this, and we got this number by subtracting 2.35 minus 0.15, which is the reduction rate we want to see. And we just kept the same standard deviation. Okay? Now, how did we define power and alpha? Alpha, we use the same alpha everyone uses, there is no question. And the power, we decided to keep a power of 90%. Why not 80? Because this is a supplementation or a supplement, and it's not very widely used, and sincerely we think it's not gonna be easy to demonstrate an effect. So we need to have a high power so we can make sure that at least we're gonna see some effects on the population. Why not a bigger power? Because this raises a lot our sample size, and we thought that, well, too many patients was gonna be hard for us to recruit. The final part, which is defining the percentage of expected dropouts, we also did a literature research, and we found two different articles that were using our same criteria. The first one was using a 20% study withdrawal rate, and the second one was using a 10% loss to follow up. The article that I just showed you and the one that we use for our definite conclusions in the draft, neither of those mention what happened or how much dropout they have. So we decided to go for a 20% withdrawal rate because these patients are taking too many medications daily and they have a lot of comorbidities, so the probability that will drop out or they will not come to follow up, it's very high. So we decided to keep this 20%. Now, how do we did the calculation? How do we actually put these numbers in STATA? So we go to STATA, we put the command that I've shown you before, and then let me just write this. We just have to put the variables that we had before. So 2.35, it's a control mean that they were giving in the article. The mean 2, which is intervention, is the one that we calculated, which is 2.35 minus 0.15, which is 2.2. And the standard deviation is the one that they were using, which was 1.42. The standard deviation for their intervention was 1.17. We decided an alpha of 0.05, and we decided a power of 90%. Don't forget it's 0.90, I know 90. And we press enter. So here we can re-verify our data. So we're having an alpha of 0.05, a power of 90%, a reduction, which is this minus, a reduction of 15%, our mean 1, mean 2, mean standard deviation for both groups. And we get a total sample size of 3,164 patients, which is 1,582 per arm. Now, to this number, we still have to add a 20% of dropouts. So we can do this in STATA. We put display, the number, just take the comma away, the number multiplied by 1.20. Why by times 1.20? Well, this is 100% and adding an extra 20% of the dropouts. So we get 3,796.8 patients. We cannot have 0.8 patients, so we will round it up. If we want to know how many patients will we have per arm, we just do this number, 3,797, because we're rounding it up, divided by 2, and we get the number per arm, which is 1,898.5, and remember we have to round it up. And this is it. This is how we do the final calculation. So here I put you guys the window that I showed you before, and here it is. Adding a 20% dropout rate, we're gonna have this many individuals, and if we divide it by arms, we're gonna have 1,899 individuals. If you want to put it in nice and pretty words, this is the way to put it. We're just putting like how we calculated, what we took the previous trial of the verralizumab, which is the one that I was showing, and at the end, this is the number we calculated, this is the effect size we were expecting, our alpha, our power, and then we add a 20% loss of follow-up, and these are the final numbers we get. So this looks very easy, but it took us a lot of time, especially because we couldn't find articles to justify this whole calculation. I repeat you guys again, this is not the one we used for our final draft. We went for the other article for our final draft, but I think was a good example to show you what happens when we change a little bit the data. So if you guys have any questions later on, you can ask me, okay? Bye guys.